Hello, hello, hello.
Hello.
I didn't take, just forgot to take in my rush to get my lunch. I need to take a picture. There we go. All right, so I think probably a good thing to do first would be to go over any questions that you have for the, oh, damn it, yeah, lab from last week, which is due tonight. I know I've had a few questions by email, and there was one in the chat earlier. So I'm just going to answer that one first. I think that was Zachary. And that was, what is xi? And so kind of whenever you write these mathematical formulas, which I'm not a huge fan of, I've got to say, I'm not much of a math person.
Professor, it wasn't what was the xi. I was wondering for the sample variance and for the standard deviation, do we replace xi with each of the numbers in the vector, or do we just do it
Correct.
with the sum of them?
So that's what xi is. I'm getting a bit of an echo going on for some reason. Xi represents all of the individual values, right? So, for example, the mean, I'm not sure if I can do a good job of that. I can't write what the mean symbol is because it's got the X with the line on the top. That's basically all of the individual values, or, sum of, divided by n. Right, so if you want to write that out in full, the mean would equal x1 plus x2 plus x3, or whatever, however many individual values you have in that vector. And that would be divided by, in this case, 3. So the I in the xi stands for the individual data points. So when you do calculate it, if it's, for example, in the variance and the standard deviation, it's the, I'll just do variance because it's a little easier. That's the square root . Hang on. No, that's not the square root. It's the sum of xi minus. Oh, that's in the Dipper. That's in the handout. So it's just like fancy way of talking about stuff mathematically. And mathematicians like to. No, not that one. I don't know why that's doing funky stuff. There you go. So let's get back. Here we go. It's got to be s squared because that's variance. So that's always presented like so, right? Which is the sum of each individual data point. Let's change that. Sorry, I'll get there eventually, promise. Right, so that's variance. Oh, fiddlesticks. Second try. I forgot that that's sum of x xi minus mean squared divided by n minus 1. There you go. That's the proper one. Sorry. Ta-da! And so really that's saying is, okay, if you have 16 different data points, then you need to subtract the mean from data point 1 and square it, data point 2, square it, data point 3, square it, 4, 5, 6, up to 16. Add all of those together and then divide it by 16 minus 1, which is 15. That will get
Okay.
you the variance.
So since the variable, so we start with 145 and then 170, then 161, and then the rest of the variables to replace the xi.
Correct. So it'd be
Okay.
145 minus the mean squared plus whatever the next one was, 170 minus the mean squared plus, and so on and so forth until you do all of them. Should be 7, I think.
Okay, that makes sense.
Yeah, it's just kind of mathematical terminology.
I also had a question.
Far away.
I'm not entirely sure how to make. I know how to make the graph, but I'm not entirely sure because you said we have to use for the standard error mean. We have to do it differently, right?
You need to use plot plus or minus 2 standard error of the mean.
Okay. Do you mind showing me how to do that?
No. Do I still have that open? I don't on that. That doesn't have standard error of the mean.
I have it on my Excel. Do you want me to go ahead and share my screen?
I'm not entirely sure how to do that for you. That's going in with one of those kind of like complicated things. All right, so are you talking about plotting the error bars or calculating the 2 standard error of the mean?
Calculating it.
Okay, so calculating the standard error of the mean. Okay, so you calculate the standard deviation by the equals and then the function standard deviation.
Mhm.
All right, this is kind of funkily laid out rather than being a 3 together, it's 3 individual points. So it's those 3, for example. Then the standard error of the mean is the standard deviation divided by the square root of the sample size. Does that make sense?
Yes.
Okay, so I've done that here. I've just actually put it in. I did figure out after the lab how to do that in Excel, but my brain wasn't working too well at that point. So that's the standard deviation, which is R3, divided by square root of 3, which is 1.74. Now to plot them, and I can open up the other file. This is just one that I had open. Basically, you go to this and like your chart, and if you go to, you can either go to design, or you can, which up here, not all Excel versions look exactly the same. And again, you don't have to do this in Excel. You can literally draw this on a napkin. And as long as you label the axes properly and it's clear, you'll get the same amount of points. No kidding, right? Just don't do it in like, you know, one of those giant jumbo markers or something, which I can't, you know, like soaks all the ink in and stuff, right? You don't have to do in Excel. I'm just showing you this because it's a useful skill to know and it might make it easier. If it ends up making it worse, then just draw it out by hand. Anyway, so you can also right click on those and that didn't actually, you need to right click on the graph. Sorry, you two, I'm going to move you over here. Does that allow me to do that? Fudge sticks. Sometimes it does. Obviously not doing this time. Anyway, go down here to error bars, more error bar options, and scroll down. And so you can calculate the plus and minus 2 SEM yourself. In Excel, all you have to do is specify the positive and negative values and it does it for you. And so if you click specify value, I've already done it because I've kind of plotted them already. But you just select the SEM, and as long as you have those in the same format as you have the things that you want to put in your graph, you're fine. And then you click on that, actually click on the next one, select that again, and then return, and you'll have your error bars. Now this is just plus or minus 1 SEM because that's just how I plot them. It's kind of like the standard for the kind of work that I do. 2 SEM is actually better because that corresponds to a 95% confidence interval. And we'll get into what all that means in a little bit, right? Because I spent some time reading up on SDs and SEMs to answer Marissa's question from Tuesday. Anyway, to do that, all you have to do is this times 2, and that's your 2 SEM. And then if you wanted to plot that instead, I'm going to change. Let's save that, and then I can just close it if I need to. Click on those. That will bring up the error bar stuff, or you can do the other way of going to design and add chart element. And I'll just show you what it looks like essentially. There's one. Hit return and hit return again. And so that's what plus or minus 2 SEM looks like. And so the reason for plus or minus 2 SEM is, actually, you know what? Before I get into that, did that answer your question?
Yes, sir, it did. Thank you.
Yeah. And so if you want to do this by hand, and that's totally fine, you just need to figure out a way of taking a picture of it to post it to Blackboard, which is pretty easy because you all got like smartphones and stuff. If you wanted to do this by hand, now you would take whatever your mean is and add that to it, right? So essentially, so if your mean is plus or minus 2 SEM, then your plus 2 SEM is, let's say that's 60, and that's...
Hey, Professor.
Hang on just a second. This may or may not answer the question. So
we can't see the full whiteboard. So can you tilt the screen up a little bit?
I'm sorry.
Thank you.
No worries. So that will be 70 because it's mean plus 2 SEM. Your mean would be 60, and your mean is minus 2 SEM would be 50 because it'd be 60 minus 10. And so when you plotted that on a graph, it would look something like that. If you're doing a dot plot, if you're doing a bar chart, obviously there'll be a bar there instead, the way I do it. So what I showed you in Excel is basically you provide the mean, and then you tell them what the plus SEM or plus 2 SEM is and the minus 2 SEM, and they calculate it for you and plot it on your graph. Okay, let me just go through the questions first. So I'm caught up. Yeah, good point about Microsoft Office, Izzy. Actually, I'd love to know how to do that because I could do with a new version as well. And I don't want to use their bloody 360 nonsense because that's a complete pain in the backside. Oh, okay, Adam. So for the graph, you need the axes, obviously. You know, the horizontal and vertical. Each axis needs a title. So in this case, you know, it's a little fancy in what you need to do, but it's just because it's for an experiment. This is what we're talking about in terms of number, like the measured variable, I guess. There's fancy words for it. I can't remember what they are right now. And these are our different treatments or classes or whatever at the bottom. Right, so in your case, because you're looking at height, males and females, it will be sex, and then you'll be male, female, female, male, whatever. Right. So you need the axes and axes labeled. You need your means plotted, and those can be as a dot plot is fine. So what I mean by that is
So are we going to be using the bell-shaped curves or the bar graphs?
well, these are discrete categories, so you can't use anything joined together because you're not looking at a continuum, even though that's a little bit of a subject open to debate. For the purposes of the lab, at least, sex is discrete. You're either male or you're female, right? Not saying that that's the same for everybody, but for the lab it is at least. So you can't join those lines together because there aren't intermediates. There isn't things on either sides of that either, right? So they have their discrete variables, and so they can't be joined up. So you can either plot them like this as just circles. And I'm just putting the error bars on so you can. No, not like that. All right, see, that's why I don't do dot plots. Anyway, they'll be up and down. I don't know why it would do that. I could go into it, but I don't actually ever draw graphs that look like that. So you could either do that or you could do bar graphs. It doesn't really matter. I think the lab handout says to do each mean as a point, so like a round circle. That's probably the most appropriate for that type of measurement, but it's not super important, to be honest. Oh, that's good to know, Valeria. Thank you for that. Or Sheets as well. I haven't really used Sheets very much. I've always just used Office, but it works just as well too. And then you need to have your plus and minus to SEM. And that's it. That's all you need for the graph. And then obviously some way of getting that graph into Blackboard. So you can print out that page of the handout and just draw it on that page of the handout, take a picture, you can scan it if you have access to a scanner. You can draw in Excel. If you're a graphics whiz, you could do it in Adobe Illustrator, you know, with like shadow effects and sparkles and kind of unicorns jumping over the top, maybe. I don't really mind. As long as you have those key elements in there. Axes labeled, your means plotted, and then the error bars plotted. You're good.
I had a quick question about the median questions on the lab assignment. It asked to enter the value to 2 decimal places, but there was a whole number that was in the middle.
So, oh, that's right, because they're an odd number of
There's
data
7.
points in each vector, then it's the middle one. So it's just 0.00. I think that
Okay.
to do 2 decimal places is just something that I copy and paste into every single math related question, whether or not the answer is actually to 2 decimal places or not.
I figured it was, but I wanted to make sure before I submitted it.
So if, for example, you had an even number of data points in that vector, then the median would be the average of the 2 in the middle. So then you would need to plot, put that to 2 decimal places. Was that the question that you wanted to ask earlier? Okay. Does anybody have other questions about the lab? Valeria, to 2 decimal places, generally speaking. So, e.g., should be this should be rounded up to, oh, sorry, bad habits. 2.36. 1 is technically 1.00. 2 point. For example. It's just because otherwise, I mean, it's in kind of biological terms, it's not really that meaningful. That's kind of where you draw that arbitrary line about how many decimal places you want. If you're dealing with very, very small values or small differences, then you need more decimal places to be able to show that difference. But after a certain point, the numbers are somewhat meaningless, to be honest. And they're just kind of visually get in the way. It's hard to see. One of the cool things, though, about Excel, let's just go back and share for a second, is that even if you, here we go. Now that's a calculated value. Come on. Anyway, so in all of these, you can change what you see to be like 1 or 2 decimal places. Like, for example, this one, you just change that using the cell function, format cells. Right. But the information doesn't go away. So if you wanted to see what actually looked like to 3 decimal places or whatever, it would still be there. It's not like it's kind of gone. So that's kind of neat. As you can see, I do a lot of work in Excel just because it's what I'm used to. And to be honest, Excel is a steaming pain in the ass when it comes to doing like some graphs and stuff. So I'd actually recommend getting used to doing graphs in better graphing software, like I think Prism, one of my former labs uses. Because Excel is just annoying. But for what I do, it works just fine. And I'm so used to doing it. Like this, for example, is a figure that's going in a paper that's getting resubmitted, hopefully in about a month's time or so. So it just takes me no effort to do, but other programs are better. Anybody have any other questions about last week's lab assignment? New? Okay. So this week should be pretty quick. And Basically, it's a kind of online version, and unfortunately, you don't really get the full benefit because it is online. You know, this is really a way of teaching you how to use micro pipettes. Obviously, we don't have any in our hands, right? So this is admittedly one of the labs where the balance between, you know, the theory and the practice is more heavily tipped towards the practice. But there's still some stuff that you can learn, and some just general kind of good practices and things like that, right? So it's, and again, it's not ideal, but it should at least give you a kind of an overview essentially of what you're going to be doing. So the idea is, this is basically the lab exercise that you would be doing, and you would be using this, right? So essentially this is a 96 well plate, so cool because it's a plastic plate with 96 wells in it, and you would be filling each of these wells with different volumes of liquid to generate this pattern, right? And basically the idea is to get you lots of practice. So unfortunately you're not going to get that, but when you do hit this in say micro or Malek or any other kind of pipetting heavy microbiology type lab, it should be a little bit easier to kind of come to terms with, right? And so, and again, this isn't going to take an awful lot of time. All of the questions that are in the assignment are based off of this PowerPoint, kind of the, you know, stuff I'm going to be talking about, and these two handouts, the actual handout that you're going to be using, right? So this is a kind of like a description of what you're going to be doing, and come on, this actual pattern, which is what you would have been pipetting out. If you come through doing GenBio 1 here, I think you do this in GenBio 1 as well, or at least you used to. And I'm certain you'll be doing it again at some point too. It's kind of tedious, so you are actually missing out on the whole boring and batshit part of it. But you won't be getting the actual kind of manual muscle memory that you would doing it. But the principles are the same. And basically the idea is that if you want to measure small volumes, you have to use things that measure small volumes accurately, right? And so depending on what you're doing, you can perhaps measure down to milliliter units fairly accurately with a measuring cylinder, let's say, if you use a small one. So you might, you know, have some accuracy between, say, oh, I'm sorry, lunch is kicking in, between say 8 milliliters and 9 milliliters, with some you know, kind of margin of error essentially. But once you get smaller than that, it's really hard work, right? And part of the reason is like the physics of water. Oops. Right, because water kind of clings to itself, right? It has a meniscus, it has surface tension, right? And so it's really quite hard because of all the hydrogen bonds between the different water molecules. It's actually quite hard to measure small volumes of water, right? And so what we use in the lab is something called a micropipette or a pipette, right? Kind of shorthand. And these are actually really cool little things. They're surprisingly simple, but very, very handy. This is what a basic pipette looks like, right? I'm sure you've seen them before. And they have some key kind of points to them, right? Essentially inside here is a plunger, right? And that plunger expels air out the front, you know, the tip of the pipette, so that when you stick the disposable tip into the liquid and you use a disposable tip because you don't want all kinds of nasty going up there, which you can't get out, and you release that plunger, it will suck up a defined volume of liquid. And then you transfer that over to whatever you're pipetting it into and you expel the liquid, and that, if you're doing it properly, will give you your defined volume. And so you can get large volume micro pipettes that go up to like 5 milliliters, but the ones that you're most likely to come across go between, say, 1 microliter and 1 milliliter. So a microliter being 1,000th of a milliliter. Now, the flip side of that accuracy is you have a limited range, right? You can't use a large pipette, say one that measures out a milliliter called a P1000, to measure out single microliters. So that's why whenever you go into a lab, you'll see a rack of 3, 4, 5, maybe different pipettes, and they each have a different range. And they each should only be used within that range because that's kind of where they're most accurate. And so if you want to measure out 8 microliters, you'd use a P20, right? Technically, you could use a P200, but you'd be outside of its range and it wouldn't be very accurate. If you wanted to measure out 300 microliters, you'd use a P1000. Because even though you could use a P200 and do it twice, like 150 each time you do that, you have the error of the measurement. And so your error kind of gets bigger and bigger. So you always have to use the correct pipette for the volume that you're trying to measure out. That's kind of a key part. Now you can change that volume within that range by using either the volume adjustment knob at the top, which is a bit you press down with your thumb, or there's a thumb wheel on the side. This is like a classic kind of Gilson pipette man type thing. They do kind of vary from brand to brand what is where, not in kind of meaningful ways, but most of them are somewhat easy to figure out. A couple are like really weird. Just try and avoid those. So you avoid it. Basically, you're screwing the plunger up or down inside that chamber, right? So you change the amount of air you can push out, which changes the amount of volume that you can suck up. Now, to figure out what's what, you look at the display on the side. That will tell you the volume that you've set that pipette to. And this is also somewhere where students get a little bit confused because there are only 3 digits. So it depends on what pipette you're using, what those 3 digits mean, right? Hopefully that makes kind of sense. It's easier to show you, but I don't have one to hand, unfortunately. If I'd thought about it when I was last on campus, I would have snagged 1 or 2 from my own lab and brought them home to show you, but I wasn't thinking that far ahead at the time. So these 3 digits measure the volume. Question 7. Give me just a moment, Dipper. I'll have a look. Make sure I'm replying to the correct question. You don't have to put units in. That's probably why it's have pitch and affinity, because you're putting in letters and it's expecting just numbers. No, don't worry about it. I mean, it's a good question to ask. Yeah, just numbers, it's fine. Okay, where was I at? Here we go. So, get that share going again, otherwise you'll wonder what I'm pointing at. So anyway, the volume that you're measuring out or you're setting the pipette to is represented by these numbers, but those numbers mean kind of different things depending on which range pipette you're working with, right? So the easiest one I always start with is the P200, right? So the P stands for Pipette Man. 200 is like the upper end of the range, right? So that tells you it can measure up to 200 microliters. If you use one of those, each digit on the side corresponds to a digit in the volume, right? So this is set, actually it's set wrong. Ha, whoever took that picture. This is set to 16 microliters, right? Which is wrong because it's outside the range. You should be using a P20. And so the first number is hundreds, second is tens, third is singles. Right, so if you wanted to make measure 116 microliters, you'd change it so that the first one read 1 and then 1 and then 6, so 116. Pretty straightforward for that one. The ones above and below it are kind of funky. And so the P20 reads only to tens, right, because its maximum volume is 20. And so here the red number is after the decimal place. So it's tens of microliters, ones of microliters, and then kind of tenths of microliters essentially. Right, so this one is reading 9 microliters because it's 090. Right, that's kind of where people can get confused. And the opposite is the case for the P1000. So this is, one is whole milliliters and then hundreds and tens. So this is reading 1 milliliter or 1.00 milliliters. So if you wanted to set it to, I don't know, say like 950 microliters or 0.95 mils, it'll be 0.95 would give you the correct volume. Now again, to change that, you can either use the, oh, that's an Eppendorf pipette. You can either use the top thing to change it, or you can use the thumb wheel, kind of the black thing there. You can actually move that up and down to lock it if you're so inclined. Some other things that are worth pointing out if you don't already know about them. You can, obviously you're supposed to put a tip on. You have a tip ejector, right? So you can fire the tip into the trash can or at your lab mate if he or she is annoying you. And also on the top of the plunger, the bit that you operate with your thumb, is the range, right? So yellow is P20 or P200, blue is P1000 for some reason, don't know why. But either way, that essentially tells you the maximum volume you can suck up or aspirate with that pipette. Now, the other key thing that you have to kind of bear in mind, and this is arguably one of the most important things to know about pipettes, is that there's 2 stops. So to suck something up, you have to expel the air by pressing down on the plunger with your thumb. But there's a soft stop, that's the volume measurement. So when you just do that with your thumb, not hitchhiking, just pressing the plunger up and down to the soft stop, that's the volume. The second hard stop, which is as far as it will go, like you can't physically force it down any further, that's used to expel the liquid. If you push the plunger down to the hard stop and then suck up your liquid, your measurements will be wrong. That's probably the most important thing to remember about using pipettes. And that's pretty much it, to be honest. I mean, this one's, to be completely frank, a little bit of a filler because we have to make the right number of labs, but also some of these things are actually trying to get you to pick up on them even though we're not able to actually do the exercise itself. right? So some kind of familiarity with these things next time you actually do need to use them will make your life a little easier. And that's pretty much it. Nothing much more complicated than that. And the assignment is pretty straightforward. It's really just talking about, picking up on stuff that I've talked about. So you have to use the appropriate volume. You can't use a smaller pipette and do multiple measurements to get one large volume. Small volumes only, below I don't know, probably below 50 milliliters, really below 5 milliliters, to be honest. And then what you're going to do for the last couple of questions is figure out the volume of liquid that you've added to the plate. This is what you'd actually do as part of the exercise in lab. And then I'll tell you in the assignment essentially the volume. It's going to be the weight, but the weight equals volume when it comes to water. I'll tell you what you actually did measure, and then you need to work out the accuracy of your measurement. And that's the last question. So this one's really not all that challenging, to be honest. You can probably bang this out in the next half hour or so. But, it'll get another one under the belt, and then we'll move on to some slightly more challenging stuff next week. So yeah, that's it from me. Does anybody have any questions about this one? I'm sure no one's going to complain about being able to get out of lab a little earlier, because my eyes are starting to point in different directions. All right, well, thanks very much, everybody. If you have any questions about anything that we learned today, particularly obviously in the lecture section, bring them with you for next Tuesday. Where are we at in the schedule? So actually, that was something else I meant to ask about. I should have done this during lecture, but I forgot, unfortunately. I don't like the idea of having an exam on the same day as you learn the material. I kind of done it like that because normally these are actually on different days, right? Because even if they're in the same week, they'll be on different days of the week. But because everything's squished in, it kind of makes a really artificial schedule in some senses. And I want you to be able to have the chance to go over what we've learned before you actually get kind of quizzed on it. So even though the exam is like available the whole day, I'd really rather put this for avoid having an exam on the same day you learn material. So that's the case for exam 1. Exam 2, I don't think it really matters because all the material would have covered on Thursday. And same for exam 3, but certainly exam 4 needs changing. Does anybody have any objections if I change these to like exam 1 to the day after? So that would be 18th, next Friday, not tomorrow, the Friday after that. And then we already talked about this, but changing exam 4, it's not really the final exam because it's not cumulative, exam 4 to July 28th.
So are you saying that the exam 1, instead of opening it on that Thursday, you're opening on a Friday and then having that 24 hours? Or are you still going to open it on? Oh, okay.
Yeah, so you still have I'll probably open it at, I don't know, 9 a.m. or something like that until midnight. But then it will just give you, the rest of the day to study the material we learned that day. Give you an opportunity to ask me any questions by email. And we'll also have that will free up that period if you want to do any recap or have any questions to ask about the material in general. The only thing is because, oh, Desiree, just give me a moment. I just saw your question. I'll answer that in a second. Because of the way the schedule was designed and because you signed up for a particular schedule, I don't want to move an exam to a day when people have something else that they've already committed to. So if you're pulling a, I don't know, like a double shift and you're working for 20 hours on the 18th, you'd probably be pretty pissed at me if I moved the exam to that day. So that's why I wanted to ask, like as a heads up, ahead of time, if anybody has any objections to that. It's somewhat unlikely, but because it's not technically in the schedule, I really need to ask you first. Cool.
Could
Well-
you just have it open up from, oops, sorry.
Sorry.
Could you just have it open up from, I guess, since all of us have different time schedules, if you could leave it open between like Thursday and Sunday?
I'd probably rather avoid doing it for that length of time, but certainly it can be from Thursday to Friday. So like Thursday after we finish class period, what would that be, like 1:30 all the way to Friday midnight.
I like that idea
Okay.
Yes.
because I do work on Fridays, but
Okay.
it'll give me that little bit of time. And I like the fact
Yeah.
that we kind of review on that Thursday, before.
Yeah, that's always a good thing to do. I try and do that where I can. It's just we don't have a lot of free time in the schedule to do that. So kind of in a way, because we're doing this online, it kind of gives us a cheeky pass on that. Okay, so I'll change the schedule to reflect that, and I'll upload that and send. Actually, better make a note. I've got a terrible memory. And I'll send that round this afternoon. So if anyone does have any issues with that, that should hopefully give them time to kind of raise those and give me a chance to fix them. Cool. All right, everybody. Study hard. Give yourself at least half an hour a week for rest and relaxation. Don't want to overdo it too much. And have a great weekend. I'll see you next Tuesday. If you have any questions in the meantime, please don't hesitate to ask. I'm usually somewhere near my laptop, even if I'm not on a teaching day. Weekends are a little bit because of other like family commitments and stuff. But I will get back to you as soon as I can. All right. Cool beans. Have fun, everybody.